Let's switch gears here and start talking about membrane transport mechanisms. We've said that the plasma membrane is semi-permeable. Some things are going to be allowed to come across, some things are going to be restricted from crossing. Permeability is going to vary by cell type and by the type of membrane. These transport mechanisms that we're going to be talking about are different. Transport mechanisms exist across all types of cells, all types of membranes, regardless of the permeability of that membrane. Transport mechanisms involve concentration gradients. Now, what do I mean when I use that term? A concentration gradient is simply a difference in concentration of some solute on each side of a membrane. So on one side there's a higher concentration, and on the other side there's a lower concentration. The membrane is in the middle. That is a concentration gradient across that membrane. That's the language that we use to describe it. Now, the mechanisms that we're going to discuss, the transport mechanisms, are three types. The first one is called simple diffusion. The second one is called facilitated diffusion. And the third one is called active transport. And all three of these transport mechanisms revolve around concentration gradients. And we'll speak about each one separately. Remember, there will be molecules that are capable of crossing a membrane simply by squishing in between the phospholipids, getting across by passing in between the individual phospholipids in that bilayer. Some molecules are capable of using the process called simple diffusion, or what is sometimes referred to simply as diffusion. Those molecules are flowing down their concentration gradient, or the other term you'll hear is with. They're traveling with the gradient. I like to use this term down because it makes more sense to me mentally that they're traveling down. They're going from a high place to a low place. In other words, molecules that cross a membrane using simple diffusion are going to travel from the side of the membrane where the solute concentration is high. They're going to go down the gradient. They're going to cross the membrane toward the side where the concentration is low. And they're going to do this by passing between the phospholipids. That's what simple diffusion is. Now, there are other molecules that are not able to cross a membrane using simple diffusion. There's something about their chemistry, and we'll talk about that something in just a minute, but there's something about their chemistry that makes it difficult for them to pass between the phospholipid molecules in the membrane. They are still going to flow down the concentration gradient. They're still going to move from the side with the high concentration across the membrane to the side with the low concentration. But they're going to do it with the help of a protein. This transport process is called facilitated diffusion. In other words, a protein in the membrane is going to assist or facilitate this kind of diffusion process. Last, there are some molecules that are not going to travel down the concentration gradient at all. There are some molecules that will cross a membrane going up the concentration gradient. And another term that you'll hear used is against the concentration gradient. In other words, These molecules are traveling from the side of the membrane where the concentration is low towards the side of the membrane where the concentration is high. These molecules are using a process called active transport. Like facilitated diffusion, active transport requires the assistance of a protein. But unlike facilitated diffusion, active transport requires a source of energy. So we have three transport mechanisms to discuss: simple diffusion, facilitated diffusion, and active transport. Simple diffusion and facilitated diffusion are mechanisms that are spontaneous. And that language refers to the fact that no energy is required to make these processes occur. They are spontaneous. What drives them, what makes them happen, is the presence of a concentration gradient across a membrane. You'll hear me say that nature does not like concentration gradients. Nature likes equilibrium. So any time that a gradient of concentration occurs on two sides of a membrane, the forces of nature are going to try to resolve it. They're going to try to get rid of it. Simple diffusion and facilitated diffusion will allow a gradient to resolve spontaneously without needing any kind of energy to be added in. Nothing special has to happen. The presence of the concentration gradient alone will make these transport processes happen. The difference between these two is that simple diffusion moves the molecule through the phospholipids. No protein is required. And facilitated diffusion requires a protein. There are two kinds of proteins that perform this job of facilitated diffusion. There are protein channels and there are protein carriers. Active transport is very different. Active transport is a non-spontaneous process. In other words, active transport doesn't just happen when there's a concentration gradient. Active transport is a process that doesn't get rid of the concentration gradient. It keeps the concentration gradient in place. It works against the forces of nature. It moves molecules up or against the gradient from the side where the concentration is low towards the side where the concentration is high. That is something that is against the natural forces. And so in order for it to occur, you're going to need to put some energy in as a cell. This active transport mechanism also requires a protein. It's a particular type of protein that we call a protein pump. So, three methods. They have some things in common. They have some things that are different about them. Now, the question is, how do you know which method a molecule crosses a membrane with? How do you know? How do you predict whether or not a molecule uses one or the other mechanism? And the answer is, it's all in their chemistry. There are three chemical features of a molecule that will determine whether or not they can cross a membrane between the phospholipids or whether they're going to need a protein to help them get across. The first factor is charge. Any molecule or atom that has a charge on it is not going to be able to cross a membrane between the phospholipids. In other words, it's not going to be able to use simple diffusion. That means only neutral molecules, only uncharged molecules are able to pass in between the phospholipids and come across a membrane. Next is size. Small molecules, in other words, molecules that have a low molecular weight, are going to have an easier time passing between the phospholipids. And this one just makes sense, right? A great big molecule would have a difficult time squishing its way in between the phospholipids to come across a membrane. Now, when I say small, I'm referring to molecules that have a molecular weight of less than 100. And we'll look at a couple of examples of size in just a moment. The last on the list is polarity. Non-polar molecules are the kinds of molecules that can use simple diffusion to get across a membrane. And that's because it's the tail region that provides the greatest barrier to simple diffusion. So since the tail region is non-polar molecules can come across it more easily. Now, I have a couple of asterisks here, and that's because polarity is not absolute. In other words, when we look at molecules, we can't say this is polar, this is non-polar. It's a relative scale of polarity. Some things are more polar than other things. Some things are less polar. Some things are non-polar. It's a spectrum, this concept of polarity. So we're going to look for generally more non-polar molecules to be the ones that are capable of crossing a membrane using simple diffusion. So all three of these things matter. It's not one or the other that will determine which mechanism a molecule uses. It's a combination of all three of these that are important in determining which mechanism will be in play. Let's take a look at a couple of examples. The first one is oxygen gas. Oxygen, as you recall, has a high electronegativity. And generally, when an oxygen atom is involved in a covalent bond with another atom, we think about polar covalent bonds. But when you have two oxygens together, as in oxygen gas, the high electronegativity is on both sides of the bond. So each one of these oxygen atoms is pulling on that pair of electrons in the covalent bond in equal and opposite directions. That means that the electron pair in the covalent bond is being shared equally. So oxygen gas is a non-polar molecule. So far, so good. You can also see it has no charge on it. That's good. And lastly, we have to ask about size. Just how big is oxygen gas? Well, the molecular weight of oxygen gas is 32. That falls below our limit of 100. So oxygen gas meets all three requirements for passing across a membrane using simple diffusion. And in fact, that's exactly how oxygen gas passes. As long as there is a concentration gradient of oxygen, a difference in concentration on each side of a membrane, oxygen gas will cross that membrane using simple diffusion. And the greatest example of this is the red blood cell or the erythrocyte, which is responsible for transporting oxygen to tissues and carrying away carbon dioxide. Next on the list are three ions: sodium, potassium, and calcium. Now, when we think about polarity, it doesn't really apply here because these are not molecules, these are individual atoms, they're ions. When we look at size, well, sodium, potassium, and calcium are all relatively small. Sodium has a molecular weight of 23, potassium 39, calcium 40. So relatively small, falling underneath our 100 limit. So far so good. But when it comes to charge, all three of these fail because they're all carrying a positive charge. Now, when it comes to charge, the reason that charge prevents simple diffusion from happening is because of the way that charged ions interact with water. Remember, water is a polar molecule, and water has partial charges within it, and those partial charges will be attracted to and will hydrogen bond with charged molecules and charged ions. So any ion that's inside of a living organism becomes hydrated with water molecules. They become surrounded with water molecules that are hydrogen bonded to them. That means even though the sodium is by itself quite small, because it gets surrounded by water molecules in a living organism, it's functionally really big, and it simply cannot get across a membrane using simple diffusion. It turns out that ions like sodium and potassium and calcium will come across the membrane primarily with facilitated diffusion and sometimes with active transport. In other words, there are going to be proteins involved in moving ions around across the membranes of cells. Water is a really interesting example because, well, we know water is polar. We know water is neutral. It has no charge. And when it comes to size, well, water's relatively small. It has a molecular weight of 18. So it's a molecule that ought to be able to cross through simple diffusion, and it does. Water can cross a membrane using simple diffusion. But water can also cross a membrane using facilitated diffusion. There are proteins in the membrane that can help water come across more quickly. Water is so critical, and the shifting of water from one side of a membrane to another is so critical that there are actually two different transport systems to move this molecule: simple diffusion and facilitated diffusion. Now, you might look at water and say, hold on a minute, how can there be a concentration gradient of water? And the answer is there can't be, because water is the solvent in living things. It's not that water uses simple diffusion as we normally think about it. Water uses a related process called osmosis to pass across a membrane in between the phospholipids. Water can also use facilitated diffusion using a particular protein channel called aquaporin. Last on our example list down here is C6H12O6. Hopefully you recognize this as glucose, another very common molecule in living cells, very important molecule. Now, when we think about the polarity of glucose, if we look at the atoms that make it up, oxygen, carbon, and hydrogen, we can predict that this is polar because we know some of these bonds are going to be between oxygen, which has a high electronegativity, and either carbon or hydrogen, which have low electronegativity. So at least some of the bonds in glucose are going to be polar covalent bonds. So it's a relatively polar molecule, so that doesn't bode well for simple diffusion. It's also a relatively large molecule. The molecular weight of glucose is 180, so it's higher than 100, which is our limit. Glucose does not have a charge on it, which is good for simple diffusion, but it fails for size and it fails for polarity. So glucose is not able to cross a membrane using simple diffusion. It requires help from a protein. Most of the movement of glucose in living cells is occurring through facilitated diffusion. There are proteins referred to as carriers that are going to move glucose down its concentration gradient using facilitated diffusion. Remember, when we talk about simple diffusion, we're talking about molecules literally crossing the membrane by slipping in between the phospholipids. That's what we're looking at in this diagram. This diagram actually needs to be read from left to right. Time is passing by as you go from left to right. You can see a piece of membrane in the middle of the diagram, a membrane bilayer. We're to pretend that this is the outside of the cell and this is the inside of the cell. And we're looking at this blue molecule. So, if we start over on the left at the beginning of the time period, we can see a very obvious concentration gradient here of this blue molecule. There's a high concentration of blue on the outside of the cell, and there is literally none on the inside of the cell. So there's a huge concentration gradient here, high and low. Now, as time passes, nature is going to want to get rid of this gradient. Nature doesn't like gradients. The forces of nature want the gradient to resolve, to go away. Nature likes equilibrium. It wants equal amounts of that blue molecule on both sides of this membrane. So we're not going to have to use any energy. The cell is not going to have to spend any of its precious energy here. This is going to be a spontaneous process. It's driven only by this concentration gradient. Now, if we assume that this molecule meets our three characteristics, in other words, it has no charge on it, it's relatively small, has a molecular weight less than 100, and it's relatively nonpolar, we know that this can happen. It can cross the membrane in between the phospholipids. It's going to travel from the side that has the high concentration, outside of the cell, across the membrane towards the side that has the low concentration, the cytoplasm. We're going to travel this way across the membrane, from outside to inside, slipping in between the individual phospholipids, going down the concentration gradient. And as time continues to pass, there will come a point where this simple diffusion process will end. And the time that it will end is when the concentration gradient is gone. This last panel shows us the moment when the concentration outside the cell and the concentration inside the cell are the same. We're at equilibrium now. Now there is no concentration gradient, so there will be no more simple diffusion. We're done, in other words. We're at equilibrium.